Hi everyone, I'm Tanya. I'm a scientist at Second Genome, which is a microbiome company based in the United States and California, San Francisco. And I'd first like to thank the organizers for inviting me. And I'm happy to be here and share our challenges in omics analysis. So just a little bit more about Second Genome, what it is and what do we do. We're a drug discovery company. It's still a small biotech company, a little older than a startup, 3 years old. We have about 15 people, and probably now is about 5 scientists since last September. And the idea behind the company is we're trying to discover drugs to treat and design new therapies for human diseases based on the microbiome science. So if you ever heard of fecal transplantation, for example, that are used for treating Clostridium difficile colitis, this is not that route we're taking. We're not trying to design new probiotics treatment that could be used to treat Clostridium difficile or other human diseases. We would like to understand the mechanism by which the microbiome either induce a disease, such as inflammation, inflammatory bowel disease, or what are specific bacteria that could reduce the inflammation. So by understanding the mechanisms by which this drives, this is our goal. So specifically, once we identify bioinformatically which bacteria species or any microbial species are associated with the disease, we then try to prove that these bacteria are indeed associated with disease by conducting experiments in mice. And then we do some functional assays as well as both in the lab and bioinformatically to identify which potentially functional genes or pathways contribute to the phenotype that we observe. And we also could test that in the lab, either like in the epithelial cell lines or other assays. And that's the whole idea. Our group, specifically bioinformatics group, started with Todd DeSantis, who was a curator of Green Genes database, which was mentioned here. It's one of the biggest database for 16S ribosomal RNA genes. We have Justin Kuzinski, who was a grad student in Rob Knight's lab, and Rob Knight's lab developed a software called CHIME for analysis of 16S ribosomal RNA genes. It's freely and openly available for the community, and it's pretty easy to use. I'm a scientist. I've joined Second Genome 7 months ago, and prior to that I did a lot of work studying the role of diet on the microbiome in humans, as well as studying the role of the microbiome in human health, specifically human nutritional status, like in obesity and severe malnutrition, as well as studying the microbiome in people living in different countries around the world, and how does the microbiome change as people age from newborns to old people. So some of the work presented about kitten microbiome was very close to me because I thought about all those questions for the last 7 years. So what we're doing is we're not really selling anything right now. Like, we don't have like a major product yet because we're mostly at the R&D stage. But we do have what we call research services, where people who don't have a microbiome expertise and who would like some help in their analysis or data generation or both, they can send us their samples, either from experiment in mice or humans, and then we conduct the sequencing and analysis and interpretation, creating a report at the end with figures and explanations, and we talk to them, explain to them what we did, what does it mean, and so it's very interactive and collaborative. And we do, for the last 2 years, we've been doing mostly 16S ribosomal RNA sequencing, as well as gene chip-based analysis, so phylochip it's called. But recently, since I joined the company, we started doing metagenomic analysis as well as RNA-seq or transcriptomic analysis. So I just thought I would show you what exactly we're doing in metagenomics, since you guys don't need a lot of introduction about microbiome, and just tell you what we're doing right now and what challenges we're facing. So metagenomics we're currently doing on Illumina HiSeq technology. So this is our example pipeline, and just briefly walk you through that. So we start with paired end read data. And then we do some quality filtering, such as removing adapters, removing poor quality bases and short reads. And there's a specific tools that we use, such as FastQMCF, PrintSeq-Lite. A lot of the samples that we're working with are host associated. They're coming from humans or animals. So we have to remove the host DNA, which is usually not very useful in our analysis of microbiome. A lot of the samples contain ribosomal RNA, not just 16S, but 5S, 18S, and it has eukaryotic, archaeal, and bacterial. So we remove that using SortMe RNA tool, and we actually save that data. We're not just discarding it, but we can use it potentially for taxonomic annotation, much like you do in 16S PCR-based assays. And then we're left with filtered sequences. Most of all of them should be high-quality microbial sequences. So what we want to know finally is that what are the functional genes that are present in this community and which bacteria are there, so which is functional annotation. So importantly, we currently aren't assembling the sequences here for a couple of reasons. First of all, as been mentioned here many times, that it requires a lot of memory, but I think for us right now, that's probably not the biggest concern, since we are utilizing a lot of cloud-based resources. And then second of all, we're working with human-associated communities, which many times, even if you have like full lane of HiSeq or 2 full lanes of HiSeq per sample, sometimes like 90% of the sequences actually map to host DNA sequences. So whatever is left is not a lot of microbial sequences to allow a high-quality assembly, especially when there's a high diversity in the sample, so many bacteria present. So what we're left is, as someone already mentioned, is the binning method, when we bin our short sequences against a database. So yeah, we are using a reference database, and we're using a biopsych. I've used in the past all my graduate and postdoc life using KEGG, and I was very used to it, and I was very comfortable with it, but now that it's not free, and for a small company like us, it's currently prohibitive to pay a license to use KEGG. So we switched to biopsych, which is freely available. It's curated. There are multiple levels of curation, from manual to more automated bioinformatic curation. And we're pretty happy with that. It's constantly updated. And they're using a slightly different schema and organization compared to KEGG. So we're using a translated also tool called RapSearch to map our sequences against this database. And at the end of this step, we get the distribution of genes and proteins present in our samples. Since the database is composed of 3,000 something genomes, we also know which genomes are present in our samples. And biopsych also has a hierarchical organization, so the genes could be grouped into pathways. So for each sample, we get all these multiple levels of information. So we have presence, absence, as well as coverage as defined by number of sequences that map to a specific feature. And all of this information allows us to do a sample comparison. So using a variety of multivariate techniques, such as principal coordinate analysis, NMDS, hierarchical clustering, identifying discriminatory organism and pathways, either just by direct comparison or more sophisticated, for me at least, models such as multivariate regression and so on. So here we also, I didn't show, but we're using also the 16S gene itself to identify the taxonomical composition of our microbial sample. And we also started using a Curtis Huttinghauer tool called MetaFlan, which gives another view on the taxonomy. So here, he constructed this database of markers that are not based on 16S genes, but based on multiple markers spread across different bacterial genomes to arrive to taxonomic annotation. So importantly, since before I joined Second Genome, they bought some software that was commercially available and did most of this stuff, and it costed like several $1,000s a year. And they paid another service to run that pipeline somewhere else. So now my goal was to replace all that software with freely available software which was performing similar or even better sometimes than the commercial. And now we start using Amazon Cloud ourselves. So if you've never used it before, it's actually, to me, it was now it is very straightforward. But if anyone has any questions on what it is and how you can use that, I'll be happy to help and explain. And the idea is that instead of deploying and building your own cluster in your lab or industry, whatever company, you can just use this virtual cloud. And Amazon is probably the biggest cloud platform you can find. And they have like this multiple types of computers that you can use, called instances. And probably now that they have a bigger computing power and it's very flexible, you don't have to invest into hardware infrastructure. You just pay by the amount of time you use. And importantly, we were able to utilize their so-called spot instances, which are 10 times cheaper than the regular price instances. So the one, like a couple of days ago, I ran this pipeline on one of our samples, which had 18 million sequences, and it took me maybe 5 hours total on a spot instance, which was like even one of the lowest powered instances, and costed me 25 cents. So I assume that if I also did a sample on top of that, it would be a little bit more expensive, but still it's not prohibitive, and anyone I think can spend 25 cents now doing their metagenomics. All right, so just also to summarize a little bit what we get from our functional annotation, not only we get some assignment of genes, but for each query sequence from any sample, such as shown here in this example, we get functional assignment for each sample. So, for example, gene GTP cyclohydrolase, and a lot of genes encode enzymes, so we get information about enzymes, like similar to CAG enzymes, as one mentioned here, and some enzymes could be grouped into pathways. And so we have a distribution, another table with a distribution of pathways, let's say LPS biosynthesis. And since we know from this, from this table, we can go back and ask if sample 1 contained a sequence or hit to this gene GTP cyclohydrolase, we can go back and identify from which specific bacterial genome that sequence came from. And thus we can get a distribution of bacterial strains in our sample as well, such as here. Again, this is all based on us using a reference database such as Biopsych, or you can use CAG. And yes, and importantly, if we have some group of samples like healthy and disease, and we see a different distribution of certain genes or pathways or enzymes, we can link this difference to a specific bacterial strain to which they come from. For us, it's very important because if we see any functional difference, at the end of the day, we want to know which bacterial strains are responsible for that difference. Or if all these functional groups of genes are distributed in all strains, then it's also very important for us to know. All right, so here's just some of the challenges that we're facing. So I start with the fact that probably because we're not using an assembly and we're dealing with very short sequences here, we are not able to map most of our sequences to a reference database. So I can't exactly tell you what is the underlying reason for that. Is it just because we have very short sequences, the nature of the database we use, the translated search tools such as RapSearch or BlastX? It's probably a combination of this. In my previous experience with different database and different sequencing technology, I could map at least 50% of their sequences to a database. And against this also will depend on the nature of a sample. If we're looking at something that is unknown, for which there's no reference genome sequence available, then we're probably not going to find it in our metagenome. Another question that has been raised here is whether to do assembly or not. And I treat it as case-by-case study. If I know that this microbiome comes from very low complexity sample and we have enough sequence reads, of course I'll do an assembly. But yeah, again, it's still questionable. So far, we're treating paired and sequences independently, and the reason for that is the translated search tools that we've used so far and tried do not allow using paired end information to map the sequences to a reference protein database. So that's why we kept them separately, which is, in my opinion, is a big disadvantage. And then for us, it's again very important to identify which bacterial strains are present in our samples and if there is any difference in representation of the strains in healthy versus disease states, for example, because we want then either try to isolate or culture this bacteria ourselves or try and find it in their public culture collections such as ATCC or DSMZ. Which database to use or whether to use database at all, such as was mentioned here. I also have some other ideas of how to do this, which is much less sophisticated than your K-MER approach, but anyways. And just, I looked up, for example, how many of the genes in E. coli genome have some functional mutation. It's something like 60%. So just using that reference as like the most studied organism, then like I wasn't surprised that I can't annotate most of my metagenome. But still, 1% seems too low to me. In some samples, and it's probably also very like case-by-case study, and some of them samples that we receive are very strange. If we use independently the forward and reverse reads, the results don't really match together. So usually you would see, if you analyze your sequences from one end and another sequencing end, they usually are identical, but in some cases they're not. And I'm not sure if it's just because the nature of the sample or something else. It's just an observation.
So what do you mean? You just reverse complement the reads and then run them again? Is that what you say?
So, so you have forward and reverse read, and when we can't really assemble them, I run them like as independent. I treat them as independent samples. And most of the time, when you look at like taxonomical profile or distribution of pathways, they're identical, but in some cases they're not. And it's, I think, mostly depends on the nature of samples that we're dealing with. So I don't know how much time I have. 2 minutes? Okay. Just wanted to give you like an idea. I wasn't allowed to show any of the results since we're startup and they don't want to share anything yet. But just to give you like an idea of some analysis that we're doing. Of course, we're doing metagenomics and comparing the healthy versus disease or other groups, representation of genes and pathways. But recently, one of the analyses I've been mostly working with is correlating human data and microbial data. So since we're interested in human diseases, one of our first interests is inflammatory bowel disease. So we have samples from healthy patients and patients with ulcerative colitis, and we have biopsy samples, not just stool samples. So we did RNA-seq on those, and we did not deplete any of the human data. So we actually have both human and microbial RNA-seq data, and we analyze them independently. And now we're trying to see are there any correlations between human and microbes, since we know that there's definitely a microbial component in causing the IBD disease, so can we identify that? So we had a number of approaches, and one of the approach that was very straightforward, very simple, nothing sophisticated, is simply to correlate the expression of every microbial genes with expression of every human genes. So here the x-axis is expression of a human gene and y is microbial gene. Every dot is a sample. And basically we're looking for correlations, either positive or negative. And I chose Spearman correlation because non-parametric, there aren't any assumptions. It was very straightforward. So I correlated literally, did on every microbial, every human, was about 23 million correlations. And to my big surprise, after doing one for any correction for the p-value, there was still 400 remain significant. And then this is just an example. Of course, I couldn't tell exactly what this human gene is, but this is, for example, one of the microbial enzymes that were responsible for peptidoglycan biosynthesis, which is a component of the cell wall, and it's a known antigen for our immune system. So here on the y-axis is each individual bar represents one sample, and the x-axis represents the expression value of that gene in each sample. So this is for microbes and this is for humans. I mean, it's not perfect, but you can see that overall there are some similar patterns. Like this group of samples that were extremely sick people, they had low expression of the microbial genes as well as low expression of the human genes. And actually it was even a subgroup here that showed some correlation as well. And there's another example just showing also the same microbial gene on the x-axis, and y is the human gene. And a lot of those could be related to immune system, specifically in the inflammasome complex. So now, like, we're going back to lab and seeing if we can actually prove some of this, our hypothesis and correlations that we found in our data. So just to summarize, I'll be happy to discuss in the next day or two our methods, some of our findings, approaches, challenges, and I'll be happy to talk about that. And yeah, thank you.
Questions?
Yes.
Tanya, a couple of questions. One of them is, you mentioned metaflam,
Mm-hmm.
which goes beyond the 16S. Can you comment, allows you to kind of nail down into the strain level, or you is still at species level?
I don't remember seeing the strain level separation there, but I think they just use, instead of just using 16S, they use like multiple markers along the genome.
Barcodes as well, additional barcode.
There aren't any barcodes. They took like a collection of bacterial genomes and tried to find like what are other markers that we can use beyond 16S.
And this last example of the IBD patients, when you get higher expression, that correlates with also an increase in copy number variation of those genes, or you are just looking at the...
We're just looking at the expression right now. Mm-hmm.
It's just a comment that you, if I understood you correctly, you haven't tried to link the paired ends
Mm-hmm.
in this, and then you found you had different profiles.
Mm-hmm.
Well, this could actually be quite to your advantage, because it would allow you at least a way to discern what is random variation
Mm-hmm.
as opposed to what is systematic. And there's all kinds of things that will be sort of sources of random variation, including, perhaps things that you may have done in the analysis pipeline that may have sort of perturbed things one way or the other. So this could actually be quite valuable to you. You get some idea of confidence that you might get from this.
I'm being cautious, and I'm just not disregarding it. I'm not throwing it. I'm not combining it. And I can see that if you have paired end and you just happen to sequence like two genes, and then one of the reads hits one gene and another read hits two, and normally you would treat it as two, but in this case they look like they're independent hits to different genes. So yeah, perhaps you're right. Mm-hmm.
When you're analysing your samples earlier on in the talk and you're looking for gains and losses, do you actually take into account the initial sample size, how much DNA you start off with, how many reads
Mm-hmm.
you've done? Do you normalise all the way through that for when you're doing the sort of correlations between
Yeah.
them out?
Yeah, it's very important because you have different amount of bacteria in different samples. So I know when they prepare the samples for library construction, for sequencing, they use the same amount of DNA from each sample. And then since, like in any case, you have different number of sequences per sample because you're barcoding your sample and you're combining them in one pool. So then all our counts are usually normalized to a number of sequences. And of course, if we do functional mapping, then we have to normalize by gene length or genome size and so on. Mm-hmm.
Have you been looking at seed as a possible database? Because that's supposed to be really good. Difficulty is that those guys don't make it easy to get hold of it. But
No.
in theory, it's open. It's an open.
It's part of M5 and MGRAS.
Yeah, but
we'll
they say it's open, so we just.
Yeah, I looked at. I think it's part of M5 and our database that they host.
Yeah.
I don't think it's updated very frequently.
I think they've got a really up-to-date version. They're just not They say they want to give it to people. Volker Meyer was here and said that everybody could use it, but you just have to.
Yeah, last time we
Give
tried it, we weren't very
Yes.
successful. I mean, I've used MGRAS in the past in grad school, and it was okay. And I understand like difference in their hierarchy. Just wasn't as easily, I think, to get to. And Biopsych was very responsive to us, and they were happy to work. And I know they update and do manual curation all the time.
So that sort of leads on to the second question. I am biased, before I should say. So, but have you used profile databases like PFAM or Interpro for the functional analysis?
No, not yet. But I'm open to ideas and suggestions of what else I can use besides Biopsych and KEGG.
Very much.
Thanks.